So I'd like to hand things back to Steve now for a moment to talk about why we're all here today, why you're here today, and what the next steps would be in enabling you folks to become early adopters of the HyperLink technology. Steve?
Great, thanks, Bill. Yeah, we'd like to get laboratory samples of HyperLink, and what we would do is we need 2 antibodies and their clone names, and that's something that we want to make sure we have exact antibodies, so the clone name is very important. And then there's a selection of colors that you'd like to put on the antibodies. So we'd like to have you, try it out in your laboratories. We'll be contacting each of you after the call to get your antibodies, clone names, and the colors you'd like to try out. Right now, if you have questions, go ahead and ask the questions through the Q&A button. Is that right, Sean?
That is correct. To ask a question over the web, go ahead and click on the green Q&A button in the lower left-hand corner of your screen.
I have a couple questions already here, Sean. I'll just start reading them off, and then hopefully we can answer them either with Bill, or I have Jim Williams here, who's the Director of R&D, with me. Can you use multiple HyperLink antibodies in one panel?
And the answer is absolutely yes. As I demonstrated to you, there is no exchange of fluorophores between HyperLink labeled antibodies. So HyperLinks are compatible with each other in multicolor panels. They are also perfectly compatible with conventionally labeled antibodies. So you can mix and match. You can use some conventionals, some HyperLinks, or you can use all HyperLinks in your panels.
Okay, I have another question here. I think you may have answered it in one of the slides. How stable is HyperLink?
HyperLink is wonderfully stable. So I've talked about an assembled probe, and what we mean by an assembled probe is an oligonucleotide conjugated antibody which has been hybridized to an oligonucleotide conjugated fluor. The unassembled components, the antibody and the fluor, are, in our experience, stable on the order of years in solution. The assembled antibodies are pretty much indefinitely stable as well, so there are no stability problems presented, no trade-offs with this technology.
Okay, we have another question here. Do you see quenching problems with high degrees of labeling?
In any fluorophore labeling of proteins, as the questioner obviously appreciates, there comes a level at which if you put any more molecules of the fluorophore onto the protein, they're going to start interfering with each other photochemically, and you're going to get quenching of the fluorescence. You'll actually get lower fluorescence as you put more fluorophores on. The HyperLink technology involves, in the cartoon that I showed, it showed a fluorophore with an oligonucleotide directly attached to it. In point of fact, fluorophores for small molecule fluors are attached to a scaffold molecule, a linear scaffold molecule, and then the linear scaffold molecule has the oligonucleotide attached to it as well. The nice thing about that is that the scaffold molecule can be made arbitrarily long to accommodate an arbitrary number of copies of a fluorophore molecule. So, ultimately, everything gets quenched if you add too many molecules. The neat thing about HyperLink, as opposed to direct labeling of antibodies, is that HyperLink is extensible because of the scaffold molecule that's employed, so that we can add many fluorophore molecules on without encountering quenching problems. So the short answer is quenching really isn't a practical problem with the HyperLink technology.
Okay, we have another question. Can non-IgG antibodies be conjugated to the oligo?
Absolutely. There's no limitation. This has been investigated. There's no limitation with respect to the classes of immunoglobulin molecules that can be labeled with the HyperLink technology.
Does the antibody need to be carrier-free?
Yes, it does. For Solulink's manufacturing efforts to oligonucleotide tag the antibody, it needs to be carrier-free. Once it's oligonucleotide conjugated, it can then be in a carrier-containing solution with no problem.
Okay, here's another question. The equal staining intensity has to do with the amount of fluorochromes attached to the oligo. I presume that the amount can be increased easily, thus allowing to make antibody staining brighter?
Yeah, this kind of touches back on the question I addressed a couple of minutes ago with respect to the fact that fluorophores are attached to a linear scaffold molecule using the HyperLink technology, and that by increasing the number of fluorophores attached to the scaffold, and thus necessarily increasing the length of the linear scaffold molecule, one can get, within reasonable limits, arbitrary increases in the fluorescence intensity of the label.
Okay. There's another question. Your slides show that the percentages of stained cells between HyperLink and conventional are the same, but it looks like the MF5 for the HyperLink antibodies is sometimes lower. Is this usually the case?
You know, in the examples that I've shown, which were comparing a HyperLink labeled antibody to the same clone that was conventionally covalently labeled with the same fluorophore, there was no effort on our part to match the brightness of the gold standard conventional antibody and the HyperLink antibody. So in some cases, with the data that I've shown here, the HyperLink antibodies were brighter than the corresponding gold standards. In many cases, they're identical in brightness to the gold standard. And in a few cases, they were dimmer than in the gold standard. But there is no systematic difference between HyperLink labeled antibodies and conventionally gold standard labeled antibodies, except I tend to have the prejudice that HyperLink antibodies overall tend to be a little brighter. But I think that's just my prejudice. The take-home message is there is no decrement in intensity with the HyperLink technology.
Oh, great. Great. Thanks, Bill. I think we're pretty much out of time at this point, and we'll get back to each of you individually if you have other questions when we're asking you about the clones and clone names, antibodies, and the colors you'd be interested in. And then we can, get you prepare your samples for you to try directly.